Great. Welcome, everybody. Uh, Thank you for joining us today. My name is Lauren Hine. I am Vice President of the A4, and I am Director of Safer Materials and Data Integrity for ChemForward, project to build a globally harmonized repository of chemical hazard assessments for safer alternatives, and also co-chair with Molly and Meg of the A4 Program Committee. And Molly here, we are working together to facilitate this meeting. Molly Jacobs is a Senior Research Associate at Lowell Center for Sustainable Production, who works with Meg and I on the A4 Program Committee. And together we're looking forward to this experiment to host a discussion group and do our best to make sure the technology goes smoothly. So today's meeting is building on a webinar that the A4 hosted on January 29th. Hopefully, most of you either attended that webinar or listened to it from the A4 website. It's available, the slides and audio are available. And it was a really, really rich webinar by two of the global experts in this field. And even to impart information at a high level took us the full time. And there was a desire to have more discussion. So next slide, please. The purpose of today's meeting is to provide an opportunity for the audience and the speakers, and with the addition of another expert, who I will introduce shortly, to ask questions and to share your experiences and expertise as well. After the last webinar, we collected questions that the audience provided, and we went through them and picked out a few that seem to be the most pressing. And we have a few questions that we will pose to the speakers, but we are open to you sharing your questions as well. So let me introduce the speakers. I would like to say that this is an opportunity for a deeper dive into new approach methodologies, or NAMs. Don't be afraid to ask anything. Our speakers have really just a huge amount of experience in the details of NAMs and also in using them in productive ways. So we want to open this to discussion. Molly will go through the actual details of the logistics, but don't be shy. So our speakers today, I'll start with Amy Klippinger. Dr. Amy Klippinger is president of the PETA International Science Consortium Limited. In her role within the Science Consortium, Dr. Klippinger collaborates with industry, academia, and regulatory agencies to promote robust non-animal tox testing approaches. Pam Spencer is Senior Director of Regulatory Product Stewardship and Quality at ANGUS Chemical Company, and we're lucky to have Dr. Spencer as our A4's inaugural president. And Margaret Whittaker is Managing Director and Chief Toxicologist at ToxServices. Dr. Whittaker is one of the pioneers in the field of chemical alternatives assessment and is a key contributor to advancing these methods around the world. Meg is a co-chair with Molly and myself of the A4 Program Committee. So Molly.
Great, thank you, Lauren, and thank you all of you for joining us today. And so as Lauren mentioned, our goal for today is really just to have a constructive discussion. So please, listen intently and participate so you can help us improve the dialogue. Not just questions here, but there's a number of people that are joining us today that have really direct expertise and experience to lend their voice and reflections on this topic. So please use this forum not just to pose questions, but also to interject insights and reflections as well. This is not your standard webinar, right? So we really formed this as a conference call meeting. So everyone here has access to unmute their own lines, but please keep your lines muted when you're not talking. And the way that we're going to facilitate this is that in your participant box, there's an option to raise your hand. So Lauren will monitor hand raisings and call on people, you know, when it's appropriate, you know, to pose your question or to offer your comment. And so at that point, just unmute your line, and we'll be able to hear from you directly. We do ask that you put your hand down afterwards, just so that we can keep this orderly. You can also use the chat box. And so I will monitor the chat box and also interject comments and questions via that mechanism as well. I find Zoom is best, you know, to use the speaker view here. I guess we thought we might actually have video of our panelists. That's not, we're just going to use voice. So hopefully the screen and the slide is taking up your full screen and it looks good and that's working as it needs to be. You probably can tell that we are recording this webinar discussion. So please keep that in mind with regard to your comments, because it will be posted onto the in a public domain on the A4's website, saferalternatives.org. So access to the slides, and we will have resource links here, will be, you know, available in the days after this webinar. Also, just at the end of the discussion, we will have a quick poll. We typically have done kind of evaluation surveys. Zoom doesn't really allow us to do that, but we will just do a quick poll to get your reflections on how this style of a webinar works for you. And with that, I'm going to turn the baton over to Pam Spencer. And we thought Pam could just do a very, very quick primer about NAMs, just again to get us all on the same page before we launch into a deeper discussion together. And so with that, Pam, please.
Great, thanks Molly, and welcome everybody. For those of you that participated in the earlier webinar, this will be a little repeat, but as Molly said, we just want to level set and make sure that we're all coming from the same perspective as we start the discussion today. So just want to review what NAMs are, and that's a term that has been coined to stand for New Approach to Methodologies, but you also hear it called predictive toxicology, Tox 21, non-animal alternatives, alternative test methods. These are all terms that have been broadly adopted, but it was really coined by the strategic roadmap developed by NIEHS for establishing how we would start to move into the use of these new technologies and approaches for chemical hazard and risk assessment. So the next slide, please. And it's coming up slow. So I just want to talk about types of NAMs because, again, a lot of times in the forums that I'm in, people think of these approaches as a single test, a test that's going to replace an animal test. And that's not really the case. There are many different tools. So I'll say there are tools in the toolbox, and these are new approach method tools that can be in silico or computational tools. So think about quantitative structure activity relationship programs. Could be machine learning. It could be high throughput exposure modeling. There's also in vitro biological profiling, and with the in vitro biological profiling, these are really meant to interrogate different parts of what we know of the biological test system or the process that we think might be occurring, the mechanism of action for the adverse outcome. And these can be cell cultures, they can be 2, 3D organotypic cell cultures, they can include things like genomics and transcriptomics, all the way to very, very complex systems that are organs on a chip that try to actually mimic the human living organism. And then this information can really then be integrated into frameworks. And so frameworks can be an adverse outcome pathway, a pathway where we know what some of the key events are in that pathway that leads to the adverse outcome, and we can conduct assays or tests to interrogate those different key events. Also in the frameworks are defined approaches, and again, these also build on adverse outcome pathways, but are hierarchy where if you're doing a defined approach, you may have to have 2 out of 3 tests that you do be concordant to be able to come to some kind of a conclusion. And then there are integrated approaches to testing and assessment, and these are IATA. And for the abbreviation, and what this is really doing is bringing together different lines of evidence. So it could be existing animal data that's out there. It could be data on a chemical analog to read across to. It could include some QSAR or in vitro biological profiling, and all of this is integrated into an assessment to decide to make a decision on whether we have enough to adequately characterize that hazard endpoint or not. And then it can actually help us determine when we need to do additional testing. So moving to NAMs doesn't mean that we would never have to do an animal test. There may be some good cases where that's the only way we can truly get an answer that we need to help inform the safety assessment. But these approaches can help us determine that and make sure that when we are moving into that direction, we're doing it sort of as a last resort. So think of NAMs as, you know, an approach and not necessarily a single assay or single model that we're using, but it's really the holistic approach of how we're going to incorporate the use of this new information to help better inform the safety assessment. We'll move on to the next slide.
So hopefully, is the next slide showing? This is Molly.
Yes,
Yes, I can see it.
Okay.
but not the poll yet.
Okay, great. So we just wanted to, before we started this discussion, just get a feel from all of you as to, you know, how you're using NAMs, or if you're using NAMs. I think this will just help Pam and Amy and Meg in the discussion to truly to know you know, how you all are working with it.
And by directly or indirectly, we mean you may use the NAMs yourself, or you may commission someone else to use them. And there's 2 questions, and you'll need to scroll for the second one to see some of the options.
And you see, we're about 20% of you voted, so we're just going to hold on a little longer before we close out the poll. A little longer. Only 50% of you have voted. Great. All right. I'm going to end it. So yeah, Lauren, what do you see here?
I don't see the poll yet, Molly. Oh, there it is. Okay, great. So 23% people said that they haven't used NAMs. 65% say they use it for hazard screening. 8% for risk assessment, 19% for alternatives assessment, and 8% for other. And of those people using NAMs, skin sensitization is 50%, skin or ocular irritation corrosion 38, inhalation toxicity 19, endocrine 31, genotox 35, and other 42. So very interesting, but it looks like hazard assessment is or hazard screening is the top use. So let's, hopefully that'll inform our speakers a little bit, or our discussants, I should say. And I'm going to kick off the discussion with a question and pitch this one to Meg Whittaker. Meg, how should we think about NAMs in an alternatives assessment context versus risk assessment or risk assessment as used in a regulatory safety assessment. And Pam and Amy, feel free to jump in after Meg concludes her comments.
Mm-hmm. That's a great question, and it's one that I'm asked a great deal. You know, in an AA, we're really trying to balance both environmental and human health considerations, as an example. And NAMs fit in quite nicely in an alternatives assessment where we need to characterize many different hazard endpoints. We're not necessarily focused, like in a risk assessment, on a single critical health effect endpoint, such as liver toxicity. And we're trying to make the right decision, generally, with incorporating hazard considerations and comparative exposure considerations. With a risk assessment or a regulatory safety assessment, we're very focused on looking primarily, not always, such as in pesticides, but in most cases on a single critical human health effect endpoint. So in some ways, it's a bit easier, in my opinion, to apply a NAM to try and identify and characterize a hazard, and then particularly using something called read across, pull that in for a critical health effect endpoint. In an alternatives assessment, you know, I want to try and ideally help a client avoid a regrettable substitution, and that includes regrettable substitutions for both human health and environmental fate and tox endpoints. So generally, we're using multiple NAMs. As you heard Pam explain, NAMs are different things. Well, we're generally picking from that grapevine and using NAMs for different hazard endpoints, and then different types of NAMs. We're using in silico protocols, in vitro protocols, and a good deal of a read across. So we're all using the same terminology, NAMs, but we're applying them differently for these three types of procedures. I don't know, Amy or Pam, if you have any input as well on how we think about NAMs in these, you know, different types of applications, or whether you agree with me.
No, I do agree with you, Meg. This is Pam, and maybe I'll just add, because, you know, those of you that know me, I'm really focused on how do we use these as we're selecting alternatives right from the very inception or of a new idea for a new product. And so I think, you know, as we look at using NAMs in that context, we are not so concerned with, as Meg said, looking for, you know, a critical endpoint that we're going to use as a point of departure for a safety assessment. We're kind of really looking at comparisons. And so we use the NAMs in what I'll call a tiered approach, that very early on we would be using computational approaches to just look at qualitatively how different new chemical candidates might compare to one another. So it's a very qualitative assessment early on. As we build that repository of information, we might move to the next step and say, okay, we need a little bit more robust information now to move that product on into the product development cycle. And we may do some in vitro tests. Ultimately, where we get to, and we have several examples of this, is we get to a product that we want to commercialize, that before we can commercialize it and it has to be registered, we will have to eventually do some animal testing in order to satisfy those regulatory requirements. So for us, it's a really nice process, because we can see, as we're using these NAMs very early on, how well they actually predict what we're going to get at the outset when we're going to finally go into registration. But we're using them in a qualitative way and not so much a quantitative way. Unless these tests have been accepted as replacements for data requirements in the registration process. So for example, with skin and eye irritation and with skin sensitization, in vitro data can replace the animal test, and in those cases, that's what we would use. Amy, anything you want to add?
Yeah, those were good points. Thank you, Pam. I would just reiterate that a lot of these NAMs do have the potential to more quickly test more chemicals and more doses of these chemicals earlier in the development process, which is a real advantage, and it can allow the development of chemicals with a reduced likelihood of dangerous chemicals either remaining on the market or ever making it to the market in the first place. So I think that's a really big benefit. And as Pam said, there are, we're seeing a lot of acceptance. We're seeing the acceptance of these NAMs increase at regulatory agencies. There's a couple nice examples, as Pam said, for skin and eye irritation and skin sensitization, as well as inhalation testing.
Great, thank you. Are there any comments or questions from the participants? Feel free to raise your hand or put a comment in the chat box if you'd like to add something or you have another question related to this topic. Don't be shy.
This is Pam. I guess I'd be curious to hear from the attendees. I mean, how comfortable would you feel using these in a qualitative sense in an alternatives assessment, and how, you know, how critical is that to really have more quantitative information? Anybody want to share?
Pam, this is Amy, and just to kill time while people are formulating answers, I think there's a really great recent example. There was a draft risk assessment of 6 commonly used preservatives published by the EPA's Office of Pesticide Programs just last Friday in the Federal Register. And it's an analysis that was conducted largely by the National Toxicology Program, NICEDM, which is the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, as well as ICVAM, which is another long acronym, the Interagency Coordinating Center for the Validation of Alternative Methods. And so these 3 groups used defined approaches based on in vitro methods to provide quantitative potency predictions that the EPA used to determine points of departure and use in their risk assessment. So this would be the first time that in vitro and in chemical assays and were used in this defined approach in a regulatory risk assessment by the Office of Pesticide Program. So I think that's a really exciting example to your point.
That's great.
Yeah, thanks for bringing that up. I just saw that over the weekend, and that is an excellent example for somebody to take a look at.
Were there particular endpoints that were used in this risk assessment that were NAMs?
So I haven't read through the whole thing, but it was so they used a defined approach to derive the EC3 values and then use that to extrapolate the dermal risk. So it was a combination of in vitro and in chemical assays and a neural network-based defined approach.
Yeah, the endpoint I believe they were focused on was sensitization. They were looking at a class of chemistries called the isothiazolones, which are known to be pretty potent dermal sensitizers. So I believe, Amy, if I recall, that it was largely focused on that.
Yes, that's right, Pam. Thanks.
Amy, we're having a request from Sherry Franjagovic. Can you actually put that link to the that publication in the chat box? That would be great if you can find that.
Happy to.
And Lauren, it looks like we have some raised hands.
Okay, let me see. Great, Kelly Grant, please unmute yourself.
Hi, Lauren, thanks. I guess one of my questions about NAMs is more in regard to using some of the toxicogenomic information and sort of how to make sense of that, you know, that maybe one chemical turns on, say, a MAP kinase pathway and another turns on some other kind of mitogenic pathway? And how do you use that in a comparative way to, or is there really a good way to use those comparatively at this point?
Well, this is Meg. I can pipe in. Well, I use toxicogenomics indirectly. For example, I've been applying guard skin and guard air, looking at, in order to try and select safer chemicals that are used in medical devices. And what guard skin does, and they license it to, it's made by Sensogen, and I have no affiliation with them. They license it to a number of labs that are qualified to run it, is they look at changes in gene expression relevant to skin sensitization. So it's an interesting way, particularly for solid items. You could use it for wearable electronics as well, to try and see, particularly for companies who choose not to perform animal testing. It's an interesting way to start looking at how gene pathways that are relevant to, in this case, guard skin, the skin sensitization adverse outcome pathway. It's an interesting way to try and evaluate an endpoint without performing animal testing. And their website, the Sensogen Guard Skin website, does a fairly good job and contains presentations. So I've used that to try and model respiratory sensitization and skin sensitization, yeah, particularly for clients that choose not to perform animal testing, or for the cosmetics industry, particularly now with you know, the law in California as of January 1st. They can't run out and perform a local lymph node assay. So that's how I'm using it. I'm not too sure, Pam, if there's some other ways you can think of the practical applications of a genomic test, but I've been very pleased with the results thus far.
Yeah. Yeah, so this is Pam, and I guess, you know, we've used it in a little bit different approach, and it's been fairly interesting. I think, Meg, what you're saying is if you know an endpoint, a specific hazard endpoint that you're looking at, you can use those and you feel pretty confident, you know, that you're getting a good, reliable comparison. We've been using it in a little bit different and broader approach. What we've been using it for is to say, can we use data from one chemical to read across from another because their toxicogenomic fingerprint or profile is similar? And we started with what we thought would be a really easy case, just two different optical isomers in the racemic mixture. And what was really interesting is that based on, because this is now we're looking at a broad biological space that we're querying with the toxicogenomics assay. What we found is that, you know, the toxicogenomics is telling us, yeah, these all seem to be equivalent and we should be able to read across. And now we've gone into some in vitro testing for skin sensitization with one of the optical isomers, and that very first in vitro assay has suggested it could be a sensitizer. So this is really intriguing to me because it gives me some perspective now on some of the limitations of the toxicogenomics, and that there's still going to need to be some follow-up, I think, when we're looking at that data. We haven't really fully agreed on how do we cover that whole biological space, and how do we, you know, interpret the data when we get it, because there are a lot of different ways to look at it. But we'll be now going into that defined approach for skin sensitization and conducting a second in vitro test to see if it confirms the first or not. If we get non-concordance between those two tests, then we'll go to the third assay. But it'll be really interesting to see where that goes. And did we just miss that difference in the toxicogenomics because we weren't really focused on it? But I think there's a lot of promise with this technology, but we still have a lot to learn with how we need to use it and what kinds of follow-ups we need to make as we do apply it. But that's just one example from my experience.
Thank you. Would anyone else like to respond to that, or shall we move on to the next question? Paul Ashford, I'll call on you next.
Yeah, hi, thanks. Yeah, coming not really with a question, but just a response to the question that was asked earlier in terms of using NAMs for alternative assessment. And I think obviously in screening terms that makes a lot of sense and allows you to be more flexible. But I think what we find, certainly talking from a European perspective, is with dealing with the regulators, certainly it's a little bit like trying to feed vegetarian food to a carnivore. They say, yeah, nice, but where's the meat? And then, so we end up seemingly, despite all the narrative which said we should be avoiding animal testing, we seem to end up back in a situation where the definitives are always still viewed that way. And I think, to my mind, there's still quite a way to go in terms of the community, of certainly the regulatory tox folks actually getting used to being able to rely on NAMs, and I don't think they're there yet. So just as a response to where I think the limitations are, that would be one.
It's a good point, Paul. I agree that it's a process, and it's one that's certainly being led by forward-thinking regulators and forward-thinking companies, because we really need that discussion on both sides. So we need the regulators who are willing to try something new, and then companies who are willing to propose new approaches and to push back a little bit when regulatory agencies are defaulting to their checklist of historically required animal tests. And one of the best examples of this that I love to talk about is for the re-registration of chlorothalonil. So many of you are probably familiar with this example where the EPA Office of Pesticide Programs worked together with Syngenta for the re-registration of this fungicide, which was a known irritant. And because chlorothalonil is a known irritant, Syngenta had animal welfare concerns. They didn't want to test this in an animal. And then in addition to that, they're aware of the interspecies differences in the anatomy and physiology of rats versus humans. So it was a good opportunity to think differently and to brainstorm with the agency on a new way to conduct this testing. So they ended up with this really elegant approach where they had agrochemical workers wear these OSHA versatile sampler tubes. They did computational fluid dynamic modeling, and this helped them to understand the deposition of the inhaled particles in a human respiratory tract. It helped them to understand the particle size distribution. And then they coupled that with in vitro testing using a three-dimensional reconstructed human tissue model, and then had an understanding of the mechanism. They had a rough adverse outcome pathway sketched out on how chlorothalonil exerts its effects. So that's how they were able to design their in vitro testing strategy. And after discussion with the agency, there was also a FIFRA scientific advisory panel meeting that was really supportive of this approach. There were some recommendations made, and Syngenta is now finishing up some of their final testing, and EPA expects to release their risk assessment soon. So I think this is such a good example. The reason I bring it up is it shows what we can accomplish when you get regulators and companies talking to each other, collaborating with each other, and maybe that approach or a similar approach wouldn't be accepted worldwide today, but if this is the first time it happens and it happens in one country, it's a process, as I said, but other countries are going to take note and hopefully come on board as well.
I totally agree. We need some thought leaders going ahead on this. I guess probably one of the big challenges from our side would be that we've got to get 27 people agreeing on that. So it's usually the lowest common denominator, but I totally agree with you. We need some good case thinking.
Okay, Irene Caldwell, did you have a comment or a question?
Yes, hi, it's Irene Caldwell from Health Canada, and I totally agree that industry and government should be coming together. I think that's a great idea. I'm hearing lots about validation on NAMs, and I'm in agreement. We need some sort of process on the validation. It seems like industry is already doing that internally, but it would be wonderful to be able to share some of that data with government and regulators so that we can see, hey, this has been completely validated, and look at the validation. We can use that data for further new substances that come in. We can use it for other read across that we have. If we end up with a database of validated processes, NAMs, it'd be wonderful.
Amy, might you be able to comment on that? I know PETA has done a lot of work to begin to pull together repositories of NAMs and which ones are validated and which ones aren't. So maybe you could address some of that question.
Sure, happy to, Lauren. And Irene, it's a great question, a great point, and I think we really can't underscore the importance of these cross-sector partnerships. I really think that's, at least in my experience, how I've seen the most progress getting made pretty quickly, relatively quickly anyway. So yes, and Irene, I'm sure you're aware of quite a few of these. There are several resources for where to get started with NAMs. There's a list that was published by the EPA. NICEDM also maintains a list of NAMs, as well as my organization. And this is just a basic compilation of really the OECD test guidelines that would be useful to get started with. But we're also seeing, of course, more fit for purpose testing, and not necessarily a one-size-fits-all approach. So I think that there has to be some flexibility in the future to, yes, rely on valid test methods. We always need good, valid test methods that we know are appropriate for the context of use. But we also need to have some flexibility in that this isn't going to be a checkbox approach. There's going to be different purposes. You know, is it a re-registration? Is it a qualitative risk assessment? What is the purpose of the study, and having some flexibility there. And as part of that, something that I think is really important, and my organization and I know other organizations have really been trying to host training opportunities and workshops, webinars for groups to learn more about these approaches, because there's a lot that's changing now. There's a lot that's new. And so we really see a lot of value in bringing people together and having discussions. What are the advantages? What are the remaining challenges or limitations? And then how do we address those?
I agree completely. Thank you very much.
Yeah. And I'll just point out, Amy had provided these links and that are in the slide deck. So you'll be able to download this after the webinar.
Oh yeah, okay.
I think, Amy, these are some of the resources that you were referring to.
Yes, that's right. The EPA's list, NICEDM's list, and then our list there are the first three. And then we have a list of webinars on our website. They're not all ours. We try to just pull from any good ones that we see online. It's meant to be a helpful one-stop shop for folks to find information. And then likewise, we have a list of companies that we know are doing in vitro or in silico testing. We try to, again, pull them all together to try to make things easy for people to find out information. And then the last two there are different training courses. One is organized by the Physicians Committee for Responsible Medicine. It's called NURA, or NAM Use for Regulatory Application. And the other is organized by the Institute for In Vitro Sciences. And this is an annual workshop. I've been to it and I love it. It's one where you actually get in the lab and can see the methods, which is especially nice for people who haven't used them before, aren't incredibly familiar with them. So it's that laboratory experience paired with some classroom learning. So it's a really good course that IIVS offers each year, each January.
Great. Thank you. Molly, would you mind going back to question 3? I think this is a really important question. It's what is being done to ensure confidence in the use of NAMs and to make sure they are being used in a consistent manner. I think it's very related to the discussion we're having right now. And I think everybody's in favor of moving away from the use of animals and toxicology and as much as possible. But the idea of validated tests provides some assurance of, say, equivalency to the regulatory required tests. But I know Amy and Pam, you've talked a lot about, equivalency doesn't necessarily mean safety, right? Because everything is a model. Even the current animal tests are simply models. But there is some concern that if NAMs aren't well enough validated to protect human health, then it could just be a cheap and easy way to say that a chemical is safe when perhaps it's not. So I'll play devil's advocate here. So there is some concern about sort of too much laxity about the use of NAMs, and what is being done to ensure that won't happen.
Amy, do you want to take it first with what's been going on with some of the validation or fit for purpose?
Yeah, I'm happy to. I don't want to dominate the conversation, but I'll do a quick start here and then turn it over to you. I'd like to start just to provide a little bit of context and back up. And I think we all know this, but we are talking about animal tests that were largely developed decades ago. I work, for example, a lot with eye irritation testing. The rabbit Dre's test was developed 76 years ago, and these tests were grandfathered in. They weren't validated for their relevance to humans or their sensitivity and specificity. And as we're doing some retrospective reviews of existing data, we're finding that while these have been used for many years, there certainly is room for improvement. So I really think of this as an opportunity to use and develop and optimize these NAMs that can really be more relevant for humans. They can be more reliable and more reproducible, which is what we all want. We all want these methods that are going to better protect humans in the environment. So I just want to say that to put it into context. When we're setting the bar, we do need to find this balance of ensuring that new methods are equivalent or better than the currently used methods, but also not creating unreasonable barriers that might prevent these really useful methods from being used. So that's my little introduction. And then I would just add to that, that there's a lot of time and resources being spent on the development of NAMs to show their validity and their when they can be used, when they're appropriate to be used. I think that through, again, workshops, webinars, official commenting periods on agency policies, scientific advisory panels, peer-reviewed publications, I think all of these opportunities and transparency in the process are really important. So I think that I'll stop there and turn it over to you, Pam.
Okay, thanks, Amy. Yeah, I think Amy raises an important point. I mean, we both participate in forums where, validation of test methods have become extremely important, in that the animal testing has been viewed as the gold standard for a long time. And there is a lot of debate going on right now that as we're looking at test methods and whether they're, I won't use validated, but fit for purpose, should we be even comparing back to the animal data and the animal models? Because in many cases, for some of the much more complex endpoints, the animal models are just not the best models for predicting. And I think, Amy, your example with the inhalation test is one where, the rodents are substantially different in physiology and from the humans. And so I think there has to be a little bit of give and take as we're looking to how we bring these new test methods to use, in that maybe it's not going to be validation like we think of validation with the traditional round robin tests across laboratories and things like that, but that there's going to have to be some kind of a hybrid. And as we are validating, we've got to start to agree on what's the right standard to compare against. And I think that's still very much open for debate at this point.
I can give one more example, Pam, just of a different way to think about this. We've been working in the agrochemical space and thinking about eye irritation of agrochemical formulation. So we know that there are many eye irritation tests, but they don't seem to be, and I'm using air quotes here, working well for the agrochemical formulations. And when I say working well, I mean that the data produced from the in vitro and ex vivo assays are not completely aligning with the rabbit data. And so thankfully we don't have human data in this case. So how can we approach showing that a new method is valid? Because we know that this regulatory accepted rabbit test, the one that was developed 76 years ago, it's not incredibly reliable. So what we're doing now is we've taken a step back and we're thinking about this through a new lens, and we're putting together this review article, working with experts in the eye irritation field and looking at human biology. So what is the structure of the human eye, and how does it differ from the structure of the rabbit or the pig or the cow eye? What are the mechanisms that take place in humans from exposure to a chemical through to your adverse outcomes, whether that be blindness or irritation or other? And using that to say, okay, thinking about this from a human biology perspective, this is what we're concerned about. We want to know what is the risk to humans. So thinking about it through this lens and saying, okay, now that we know what's happening in humans, what models best replicate that? So we're coming at it from a different approach of not just aligning, does this animal test 100% align with the in vitro test? Can they predict the same exact categories? I don't think that's, in a lot of cases, ever going to work perfectly. So I do think we need, again, a way to show that they're valid, but a different way to think about it. And I do think that this is one way to tackle that question.
Great, thank you. Kelly, Moran, would you like to, you can see in the chat box, everyone, that Kelly's made a comment. Would you like to say something, or should I just share your comment?
Oh, this is Kelly. Can you hear me?
Yep.
Oh, okay. Yeah, I just wanted to point out OPP's been using these EPA's Office of Pesticide Programs has been using in silico NAMs for a while. And I actually, part of why I'm interested in this whole conversation is that I'm really trying to figure out whether they're better than nothing and how they're using them is appropriate. And I've been very focused on the ecotox side, which is not what we're talking about right now. But that usage isn't novel, and it's kind of selective how they're using it. So I've been trying to figure out what's going on. So I'm curious if anybody has insights or if anybody else is following the pesticides arena. I call them pesticides and not agricultural, just as agrochemicals, just as a side note, because more than half of all pesticide use is in urban areas, and that's accelerated a whole lot in the last couple months, as many of you may be aware.
Would anyone like to address that? Meg, maybe that's an area you've worked in with the ecotox.
Well, it's interesting, the article that I mentioned in the chat box to everybody that goes over establishing ground rules for in silico toxicology protocols, and it sounds like it's great it would be great then for PETA to prepare a similar one for in vitro toxicology protocols. So the article I mentioned by Maya et al. Mentions and identifies that there are 15 specific uses of in silico toxicology protocols to address certain endpoints, including or applications, including ecotox, endpoints. And it's important as we start, I think we've come a little bit further along with in silico. And the article's interesting because it has many competitors who are working together to develop, look, these are the ground rules. You have to go through this process to make sure that it's an appropriate protocol. Is it reliable? Are you showing everyone what's under the hood, just like the risk assessment community did? Well, now in the in vitro setting, we're going through this too, because I've seen a lot of people knock on my door and they've run out and they've worked with certain labs that don't have as much expertise in vitro tox, but they're running some assays, and the data aren't very useful and don't answer all of the questions we need answered. And I think it's important that, especially for agrochemicals, that we think about the limitations of the tests that are there, or if they're running in vitro tests, what questions can the data answer? Because I don't think there's as much consensus with the in vitro test protocols that are out there as there are in silico, although there are limitations there too. And then to again emphasize, a lot of people are afraid of NAMs or in vitro testing. And as we move away from in vivo testing, we'll often have an eye irritation assay from 40 years ago that's based on 3 animals. And it's only partial data. And it's kind of like the scarlet letter. I was recently looking at a chemical, and I can't prove it, but I know that eye irritation assay wasn't run appropriately in the 50s, and it's just given this chemical a bad rap. And so now we're running through and trying to figure out, using in vitro testing, how can we try and disprove a study from the 50s in 3 rabbits, and which isn't really been relevant for humans. So I loved Amy bringing that point up, that the gold standard isn't always in vivo testing if it's in a very small number of animals, and the test, all the test data aren't even there for us to work with as toxicologists. Then it makes it a little bit easier to let go of that and say, well, let's look at what we can do from a NAM approach. And it's nice to see that, 10 years ago, that was a hard argument to make with clients, but it's getting much easier now because of organizations like A4 and PETA, and then people like Pam practically applying it in the corporate world.
Thank you, Meg. Well, I think I'm going to stop the questions here. And Molly, if you could jump 2 slides ahead. The next one is the resources, and the next one after that is some upcoming events, webinars and meetings that are occurring. Thank you to our speakers for providing this list. Again, this list is available in the slide deck that will be available on the A4 website soon. We have just a few minutes left, and I'd like to, next slide please, to ask you all, the people who have participated here, and I mean, we've had some great discussion, and this is such a rich field. We've been thinking about whether or not A4 should form some kind of affinity group on NAMs to continue this kind of discussion. We could use it as a place for exchange on challenges and experience. We could work together to outline a framework for how to use NAMs in alternatives assessment. We could advocate for or offer training in NAMs. Would there be interest in this group for ongoing discussions? We could continue on with periodic meetings like this. We hopefully, the next slide, Molly, I'll jump ahead a little bit. We will be hosting a virtual symposium on alternatives assessment. The meeting was planned to be in person in California, but we're going to host it virtually in the fall, and the program will be announced soon. But as part of ongoing symposia and as part of the virtual symposium, we could have a working group on NAMs. So would anyone like to address that, and any thoughts on what might be, what A4 can do to help promote NAMs and to promote all of our understanding and use of them in the most effective ways.
Why don't I launch a poll just to tap people's interest, and we can have a discussion around it.
Okay.
And while we're waiting for those results, if I again can just kill some time here, one of the webinars that you showed on the screen was an EPA Office of, it was a EPA Office of Pollution Prevention and Toxics webinar that is being held on Wednesday. And they will be sharing, for those of you who are interested in skin sensitization, they'll be sharing an update on the submission and acceptance of some of the defined approaches for skin sensitization testing. So I just wanted to highlight that because I know regulatory acceptance is really this key topic. We can talk all day about the methods that have been developed, but without that piece, you know, we're not moving forward. So if anyone's interested in that, I did want to highlight it.
Thank you. Okay. It looks like almost 60% of the participants would be interested in participating in an A4 affinity group on NAMs. 8% probably not, and 33% maybe. So we can have, don't have much time for discussion, but if you would like to put anything in the chat, or if you could send an email to Molly or myself, Lauren at Kemp Forward, or Molly at the Lowell Center. What is your email, Molly? We don't have it in.
I can write it in the chat for everyone.
Okay, thank you. And I'm just Lauren at chemforward.org. Let us know what you might be interested in participating on. We could pick a specific endpoint. We could, whether that be ecotox or sensitization or something like that, we could compile, work together to compile resources. We could come up with a framework for how to use NAMs in AA. So all good ideas would be welcome. Feel free to follow up. It just seems like this is such a ripe topic, and whether you're a practitioner or not, it's still of interest to a lot of people. Would anybody like to offer a thought before we need to wrap up?
This is Pam. I'll just say thanks for the feedback and stuff today and the discussion. I thought it was really helpful. And, you know, the more we can understand what's needed in the AA front, I think, as Lauren said, you know, with an affinity group, we could certainly move some of those initiatives forward. So really appreciate the feedback today.
Great. And hopefully you enjoyed this format as well. It was definitely experimental on our side, and I feel like we really did have some good discussion, even if we can't be in person. So thank you. And last slide, I'd like to encourage any of you who have not yet joined A4 to join. It's not very expensive, but you would be supporting a new professional association, and the only association dedicated to advancing the science, practice, and policy of alternatives assessment and informed substitution. We work collaboratively. We take on whatever projects and educational opportunities are of greatest interest to our members. So I'd like to thank you all for joining the discussion today. And don't hesitate to send us any thoughts or feedback you have once we end this meeting. So thank you, stay well, and thanks very much. And it looks like there's a last poll for Molly for a little feedback.
Thank you.
Thank you all. Have a good one. Thank you to our speakers. Great job. Meg and Pam.
Have a nice day.
Bye.